Welcome to this public talk, What Lies Beneath, Views from a Global Fleet of Ocean Robots. My name is Katja Fennel. I'm a professor in the Oceanography department at Dalhousie University in Halifax, Nova Scotia. This is the public version of a talk. A few weeks earlier, I gave a more scientifically focused talk that you can also find in the CMOS YouTube feed. So if you're scientists, I would recommend that you maybe tune in to the scientific earlier version of this talk. So this is about ocean observation, specifically autonomous ocean observation using robots. And I'm going to talk about the motivation for why this is needed, and also sort of the opportunities that this new mode of ocean observation affords us. And so the need for better ocean observation, which I'll spend a bit of time talking about, is really rather pressing. The ocean is warming, it's becoming more stratified, and its circulation is likely changing. You all know about global warming. What's important to point out is that actually global warming is primarily ocean warming. So the overwhelming majority of the heat that is added to the atmosphere, ocean, global system is added to the ocean. And that's shown in this graph here. The x-axis spans 40 years. And these different colored areas indicate the amount of heat in various components of the Earth system. And the light blue and dark blue ones correspond to the upper ocean and the deep ocean. So you can see that the majority is in the ocean. It's an enormous amount of heat. It's actually equivalent to about 5 Hiroshima-class nuclear explosions every second for the last 25 years. That's how much heat has been, additional heat has been absorbed by the globe due to greenhouse gases. And that leads to ocean warming and also increasing stratification of the ocean. So this graph here is a 20-year time series of profiles of ocean temperature in the upper 2,000 meters of the ocean. And they're actually plotted as anomalies. So that means the average temperature or the average profile is taken over this whole time period. And then the actual temperatures are shown as deviations from that average temperature. So the average is sort of reached somewhere in the middle of this time series. And you can see that the ocean started out cooler than average initially and is now much warmer, especially in the surface. Yeah, so the surface warms most quickly, which makes sense, but also the subsurface, right? 1,000 meters, so the first kilometer below the surface is clearly warming, and we can even see it below, right? In the lower 1,000 meters of this top 2,000 meter slice. And that means that the density stratification of the ocean is also increasing on average, which has all kinds of implications. So the ocean is not only taking up a lot of heat and mitigating basically warming in the atmosphere by doing that, it's also taken up a lot of CO2. So about 30 to 50% of the anthropogenic, so the man-made CO2 that we have been adding to the atmosphere since the Industrial Revolution has been taken up by the ocean, and that mitigates global warming too. This graph here shows the water column integrated amount of this man-made CO2. So you can see that's primarily taken up in the North Atlantic, which is where the surface ocean and the deep ocean are connected most directly through the formation of deep water. So it's sort of an efficient transfer route to get carbon from the atmosphere into the ocean. Another region where this happens is in the Southern Ocean, but the largest inventory of this man-made carbon clearly is in the North Atlantic. So in a way, that's a positive because it mitigates global warming, but it also leads to what's called the other CO2 problem. And that's referring to ocean acidification. So when CO2 dissolves in seawater, it affects the pH balance of seawater and basically pushes it toward more acidic conditions. And that, of course, has implications for organisms that use calcium carbonate as part of their structural body parts. Coral reefs come to mind, right? They are affected by warming, by acidification, but also all kinds of other organisms are negatively affected by this. The ocean is also losing oxygen, shown here in the bottom left panel. So this is a global map showing the rate of oxygen decrease in the subsurface as percent oxygen, dissolved oxygen per decade. And you can see that it's, so the red colors indicate negative, so decrease. It's red pretty much in large paths of large swaths of the ocean. And the rate of change is actually quite remarkable, -2% up to -4% per decade. That's actually pretty surprising that given the relatively sparse observational data set we have available, we can find this really clear decreasing trend already. And the whole process of global warming is just beginning. So it doesn't bode so well for the oxygenation of the ocean. And of course, oxygen is needed by all marine organisms that get their oxygen from the seawater. Marine mammals come to the surface and breathe air, but they are the exception, right? The other marine animals do need to take up dissolved oxygen from their surrounding seawater. So that's a concern. Also declining is the amount of phytoplankton in the ocean, or plankton in general. And so phytoplankton, they are microscopic plants that photosynthesize and basically form the base for the marine food web. And it appears that they have been declining. So this graph here on the bottom right shows an analysis based on a really long time series of observations or data set that's based on Secchi depth observations. So it's a very simple measurement. It's basically a white plate on a rope that is lowered over the side of the ship. And so the operator will lower it until he or she can't make out that plate anymore, and then record how long that rope is. Yeah, so it's basically the depth to which you still can see that white plate, or the depth where it stops being visible. Very simple measurement. And because it is so simple, it's been a measurement that's been made for a really long time. And we have this long data set and are able, like 100-year long data set, basically, are able to say that the clarity of the water seems to be increasing, which is an indication of plankton communities declining. And that, of course, is not a good sign for primary production, right? Which, as I mentioned, forms the basis of the food web. Primary production also fuels the biological carbon pump. So that's the vertical export, so export to depth of organic matter that's created photosynthetically in the surface ocean. And that's a really important route for sequestering atmospheric carbon in the ocean. And of course, the marine food web depends on this primary production. So lots of changes going on. And this is just the tip of the iceberg, really, because as I will explain more, we don't really have a great set of observations to document these changes. All of these are based on relatively limited observational data sets. So what we're seeing is the tip of the iceberg. There is much more happening below that we don't see. We sort of know it's there, but we really need to start looking beneath the surface, getting beyond just looking at the tip here to understand what's going on. And that's sort of what this talk is about. And so I want to start by sharing a quote here that the late Walter Munk said in 2002 when he was 85 years old. He actually lived to over 100. He recently died. But so he was a pioneering oceanographer, really well-regarded, well-respected, early proponent of ocean observation. And he said this, sort of at the end of the 20th century: sampling is such a dull word. Nevertheless, the concept is of immense importance. Most of the previous century, the 20th century, could be referred to as a century of undersampling. So undersampling, by that he means inadequate, insufficient sampling of properties in the ocean. And that's, let me sort of elaborate on that a little bit more. So let's sort of talk about what there is available in terms of global, well-resolved, sustained observation programs of the ocean in the 20th century. I'm just going to list them here. So we have a really long record of tide gauges. So in every harbor city, somebody is responsible for measuring the sea level elevation a few times a day. And that's a really long, expansive global data set, really useful data set. And then we have satellite observations, sea surface temperature, sea surface height, and ocean color, all three from satellite, have been transformative, have really opened our eyes to a lot of the processes that are going on, how dynamic the ocean is, showed us that there were a lot of misconceptions actually in place before these observational programs came into effect. But they're not enough, right? Ocean color in terms of biological and chemical properties is not enough. It's a really important data set for sure, but it's not sufficient. So then 21st century, there was one really additional important observation program that came into effect. And that's called Argo. And it basically consists of a global array of profiling floats, ocean robots that autonomously profile the ocean. They move up and down, measuring temperature and salinity. In the upper 2,000 meters, there are about 4,000 of them distributed evenly, and they have been for about 20 years. They have been making these measurements. And in fact, this graph I showed you on the first slide that showed the increase in temperature in the upper 2 kilometers of the ocean is based on measurements from this Argo array. Hugely influential and important program. Just to give you an idea, in just the period, the 16-year period from 1999 to 2015, these floats measured 1.5 million profiles. All the previous profiles that are available over the past 100 years, measured from ships, amount to only 500,000. So in 16 years, this program was able to make 3 times as many measurements and had been available before as basically have ever been made. And they are doing it at a relatively cost-effective way, and we get the data in real time. So really amazing program. And I want to talk a little bit more about how this works actually, how these floats work. So there's a picture down here showing you one of these floats in the water. There's an antenna sticking out. So it's relatively easy to deploy them. They're just thrown overboard essentially. And then the way they work is they're able to regulate their own density. They can essentially make themselves heavy and make themselves light. So when a float is deployed, initially it will make itself heavy, sink to about 1,000-meter depth, and then drift passively for about 10 days. After 10 days, the clock in the float tells itself to make itself first a bit heavier yet, to sink down to 2,000 meters, and then rise to the surface. And on this rise to the surface, properties will be measured, temperature and salinity in this case, and then transmitted via satellite once the float has reached the surface. So the data is pretty much available right away. And then the cycle continues. The float again sinks to 1,000-meter drifting depth. And sort of a little teaser here. Maybe you can think about how this might work. The float makes itself light or makes itself heavy, but it doesn't change its mass. So maybe at the end, we can briefly talk about how this is possible. I'm thinking here about a potential sort of audience of high schoolers. So that's something maybe that you can think about in the back of your head. Okay. So this 20th century undersampling is actually worse for biogeochemical properties compared to physical properties, right? I've talked about temperature and salinity, talked about sea surface temperature and sea surface height, as well as the measurements we have from tide gauges. So those are all measurements that allow us to better understand ocean physics. But for ocean biogeochemistry, it's much more sparse. Ocean color really is the only one we have, and that's not enough. And I'll show you a little bit more why that is not enough in a moment. Now in the okay, of course, we also had measurements from ships. And so on the left here, I'm showing you the amount of profiles measured every year starting 1965 from ships, either through CTD casts in red or bottle casts in yellow. So those are all the profiles available in the World Ocean Database. And I'm showing this for oxygen, chlorophyll, and nitrate because those are the three best observed properties. And what you can see is that actually the number of available profiles has decreased over the last few decades, right? Actually, oxygen has gone down since 1990, like the total number of available observations. Chlorophyll has been going down for the last 15 years, and nitrate has been going down since the 90s as well, which is kind of surprising. You know, you could guess that by just looking at that maybe we don't have open questions about the ocean, when in fact the contrary is true, right? We have all these big changes that are concerning that we don't understand well, and at the same time we're observing less. So there's a real need for observations. In the 21st century, there was one additional observational program that started that is relevant to ocean biogeochemistry, SOCAT, Surface Ocean Carbon Atlas. So this is a very successful program where surface ocean carbon variable, the partial pressure of CO2, is measured autonomously or unattended from ships of opportunity. So basically commercial vessels have agreed to take on the sensors necessary to make those measurements. And that's a very useful program for sure, but it's also limited, right? It's limited to ship tracks where commercial vessels go. And it's only one variable in the surface of the ocean. And that brings me to one other thing, which I'm really excited about, right? So this is really what this talk is about. There's the prospect of a biogeochemical Argo program, or BGC Argo. I'm going to tell you more about that in a moment. But first, I'm going to elaborate a little bit more why this sort of this lack of biogeochemical observations is a problem. It prevents us from gaining really a deep understanding of the processes that we think are important. It prevents us from assessing the current ocean state and from predicting how it will evolve in the future. So I'm going to take large-scale biogeochemical models as example, and I'm going to show you just some of the evidence for this assertion that I'm making here. They are at present not properly validated or constrained by observations. Just satellite chlorophyll is not enough. And so what I mean by biogeochemical models really are the ocean equivalents of models that are used for weather forecast, right? So our weather forecasts are really remarkably good these days. Yeah, we trust the weather forecast for sure. It's based on predictive models that ingest a lot of observations about atmospheric variables. And that's why they are so good. The situation is different for the ocean. So first, this is an illustration here of why satellite chlorophyll is not really enough to help us understand what's going on in the ocean with regard to photosynthesis and productivity. What I'm showing here is the comparison of chlorophyll measurements from satellite. They're shown here in the Gulf of Mexico. So picture yourself here standing on a high hill somewhere in Canada and looking southward. So we're looking at the Gulf of Mexico here. This is Florida. This is the Yucatan Peninsula. And in color here is the chlorophyll concentration, where blue corresponds to very low values, red to high values. So you can see that chlorophyll is elevated near the Mississippi River Delta, because a lot of nutrients are being discharged by that river. So that's not surprising. But in the open Gulf, it's very low. And then what you can see below here, these vertical colored lines, those are profile measurements from Argo floats that had fluorescence sensors, so sensor that can measure chlorophyll. And so the point here is that most of the chlorophyll is actually well below the surface, right? The satellite can't see all this chlorophyll here. The satellite sees only the surface where the concentrations are very low. The maximum really is more between 80 and 100 meters. And that is true not only for the Gulf of Mexico, but for the global ocean. Large parts of the ocean have the majority of chlorophyll well below the surface and not visible to satellites. And so let me talk a little bit about sort of why I'm making this statement that models are not properly validated and constrained. I'm going to show you two examples here that use Earth system models. So those really are state-of-the-art global models. Many nations have their Earth system model that they use for climate projections, for the Intergovernmental Panel of Climate Change. And this here is from a paper that came out in 2012, looking at Arctic primary productivity and showing the evolution of Arctic productivity, so averaged over the whole Arctic, essentially, in 3 of these Earth system models. The gray one here is the US model, the green one, the German model, and the purple one is the French one. And so they are shown as anomalies. They're forced to agree in this period from 1980 to 2000, so that we can focus really here on looking whether they give a similar forecast or diverging forecast. And what you can see here from 2000 to 2100, one model indicates a drastic increase in Arctic primary production, one model a moderate increase, and one model a decrease. So largely diverging futures being predicted here for the Arctic in terms of primary production. And these are just 3 illustrative examples from a larger group of models. In fact, there are 11 models that were used here. And this gray bar here shows you the total range from these 11 models. Right? So there's even another one that goes lower. And so, you know, what that tells us is that we can't really trust these individual model predictions much. There's a lot of uncertainty in predicting forward. So now I'm showing you another comparison of Earth system models. This is the annually averaged surface nitrate concentration in the North Atlantic. And this is for the present. And so there's this grid of 9 models here on the left. And then the plot in the upper right is based on observation. So this is our best guess of what the truth looks like. And what you can see just by visual comparison is that really none of the models represents the truth particularly well, right? So it's not only the predictions that are uncertain, also the present state is not represented very well by these Earth system models. And you know, the reason for that is really that we don't have enough observations to properly validate and constrain and even formulate the models. And so, you know, at this point, maybe I should remind you or tell you, in case you didn't know, that I'm a biogeochemical modeler, right? So that's my specialty. I am convinced that models are extremely important and useful tools, that we need them, but we need to get observations that help us make them more useful and more reliable. And I'm not the first person or the only person to say this. In fact, it is something that I've heard from really early on when I started my career, including from scientists like Carl Wunsch. So Carl Wunsch is an oceanographer at MIT who's dedicated large parts of his career to improving models. And he said in a paper in 2016 about this process of moving from just modeling to more observation-informed model estimation, he said the following: complicating this transition has been the rise of the ocean/climate modeling culture, in which computing over long periods and comparing models are easy compared with making measurements. I do not attempt to evaluate the large and contradictory literature on unconstrained model calculations of possible ocean change. So in a way, he's referring to exactly what I've shown on the previous two slides, right? We are running these models for long periods of time. It's relatively easy actually to run them now with the powerful computers that we have. It's hard to make measurements, right? It's expensive. It's not something that leads to instant gratification. It's sort of painstaking work, not glamorous, but it is important, right? Because models without observations really are not that useful. And so there's one other quote that I wanted to share with you. This is by my colleague Marlon Lewis, here, a colleague of mine in the Department of Oceanography at Dalhousie. He's an emeritus professor now, and he's somebody who loves to go to sea, right? He often would quote Red from The Wind in the Willows, describing going, there's nothing better in life as messing around on a boat. I'm not exactly capturing it, but that's sort of the gist of it, right? He loves to go to sea. Yet he said in 2017, I no longer believe that manned missions to sea are the most effective way to tackle the most important problems in oceanography. Yeah, so it's still important to go to sea. We need ships, we need oceanographic vessels, no question. But in order to address these problems, that some of which I've shared with you at the beginning of this talk, we need better ways to measure the ocean. And that brings me to what I've already mentioned earlier, alluded to, the Biogeochemical Argo program. So an extension of the existing Argo program for physical properties. And let me tell you a little bit more about it. So basically the plan is to add more sensors to these Argo floats. 6 additional parameters: oxygen, nitrate, pH, chlorophyll, suspended particles, and downwelling irradiance, so light, basically. So 6 additional parameters can be measured on these floats. All of them are really important parameters that we should measure. So that's indicated in the second column here in the table. They are all identified as what's called an essential ocean variable or an essential climate variable. So they've been identified as the most important variables we need to measure in order to address the difficult questions that we face as society in the context of global warming. And so the float array makes it possible for the first time, really, to make sustained and cost-effective measurements of these essential properties. It's unparalleled in terms of the spatial and temporal coverage that we can obtain, and it doesn't have biases, right? No regional biases, no seasonal biases. The data is available in near real time, and it is freely available to anybody who's interested in it, right? So anybody can download this data. And so the plan for the BGC Argo array is to populate the ocean with 1,000 of these floats, in addition to the floats measuring the physical properties. And so if we assume that a float lasts for 4 years, that means adding about 250 floats per year, which at a cost of $100,000 per float amounts to 25 million per year, which might sound like a big number, certainly to a private citizen, but in terms of ocean observation and global programs, it's really remarkably cost effective. It's much, much cheaper than measuring by ship. By ship, it's impossible to obtain this coverage that we can obtain with floats. And so it's really a rather cost-effective program. It's also quite cheap compared to satellite missions, which are really, really expensive. So let me tell you a little bit about the status of this BGC Argo program. So this global map here shows the locations of active BGC floats at the end of last year. The different colors indicate different combinations of sensors. So most of them don't have all the 6 sensors yet that I showed on the previous slide. There are many that have 5 sensors, so they're either missing light or pH. 4, 3, there are a lot of them that only have 1 sensor, oxygen. So oxygen was the first BGC sensor that started to be put on these Argo floats, and it's still sort of the most widely spread biogeochemical measurement. And this graph here shows the number of these profiles that have been collected over time. In blue is oxygen, so you can see it started first and ramped up the fastest and is the most measured property still, followed by chlorophyll and backscatter. So those are measurements that allow us to get insight about plankton communities and productivity. In red, we have nitrate. It's the most important nutrient to plant growth in the ocean. Light blue is light. Of course, light is essential for photosynthesis, so understanding its penetration in the surface ocean is really useful. And pH is sort of the most recent addition to the sensors, but of course really important because it informs us about inorganic carbon and ocean acidification. And so how does this number of data compare to the traditionally collected data? So here's now another version of this plot that I showed you before, right? Number of profiles measured over time of these 3 properties through traditional means in red and yellow, and through Argo in blue. And you can see that in just these last few years, even though we're not even close to reaching the target of 1,000 floats in the ocean, Argo has already become the most important source of observations for all 3 properties. So my guess is that at present we are maybe at 16% of this target of 1,000 floats with 6 parameters, right? Guesstimating from this graph here. At about 16%, Argo is already providing 60 to 80% of the total available observations. Obviously, this will increase as we get closer to the target, the global target of 1,000. But already these observations are providing great insights. I'm going to give you 2 examples. This one is from a publication by Amanda Gray, a young professor at the University of Washington. And she looked at floats that were deployed in the Southern Ocean and was interested in the carbon uptake of the Southern Ocean, so uptake of CO2 from the atmosphere. And so the conventional wisdom was that the Southern Ocean is the largest sink of atmospheric CO2 in the global ocean, right? It takes up a large amount of CO2. Even though data coverage, as you can imagine, is somewhat sketchy in the Southern Ocean, right? There are no commercial vessels, or very few, certainly not when it's winter in the Southern Hemisphere. And also very few ships would go all the way to the ice edge here, the southernmost parts of the Southern Ocean. So Allison and her colleagues took float measurements so that the float trajectories are shown here by the colored dots. And the colors correspond to different zones of the Southern Ocean. And then based on these measurements, she estimated the air sea flux of CO2 and compared it to the previously available measurements. And that's shown here in this graph. In black are measurements from a paper in 2009 by Taro Takahashi. So this has long been the gold standard of air sea CO2 fluxes. Then in light gray are measurements from the Global Carbon Project. So this is, and this particular one was published in 2017, so that's updated every year. That's sort of the best, most recent available estimate of air sea CO2 flux. And then in color are the fluxes that Allison and her collaborators estimated. And so you can see that they agree reasonably well in the subtropical zone, in the subantarctic zone, even in the polar frontal zone. But then in the Antarctic southern zone here, the southernmost zone was sort of open ocean, open water. The previous estimates completely missed that there's a large outgassing of CO2 occurring in that region. And so that basically missing this really large outgassing of CO2 meant that the uptake of CO2 in the Southern Ocean was basically estimated to be an order of magnitude too large. So based on Allison's work, we had to revise downward the magnitude of Southern Ocean carbon uptake by an order of magnitude from 0.8 or 0.9 to 0.08 petagram of carbon per year. So this is also, in my mind, one example of sort of this human nature, right? We see the tip of the iceberg. We should know there's more to it, but if we don't see it, we tend to sort of pretend it's not there or like to think it's not there. And that's apt in this case because this outgassing of CO2 in the southernmost zone really shouldn't be a big surprise, because this is where water upwells, deep water upwells from the deep ocean to the surface. Whenever that happens, there's usually outgassing of CO2 that goes along with it. We really should have guessed that that's the case, but somehow instead, we didn't have measurements. We pretended that's not going on, and instead have this big uptake flux that's compensating partly for this outgassing in these more northern regions of the Southern Ocean. And this is a second example here. Now, this is about organic carbon fluxes, specifically the vertical flux of photosynthetically created particles in the surface ocean, from the surface ocean to depth. So this sort of is relevant for the biological carbon pump, which I mentioned at the beginning. And this again is based on float observations. In this case, it's backscatter observations, so observations of particles. And shown here are events recorded by two particular floats, one here in our region in the Labrador Sea and one in the Southern Ocean. And the float measurements were processed so that we get a proxy of large particles, right? So larger particles of organic matter and small particles. And what you can see here is really there is an event following a bloom. So we have a bloom in the surface, algae grow exponentially, and then there's an event here following that bloom at the tail end of the bloom, where a lot of this organic matter sinks to depth and does sink relatively quickly. And a similar example here from the Southern Ocean. And so Nathan Briggs, the lead author of this study, and his colleagues used these measurements to look into one sort of big puzzle related to organic matter flux in the vertical direction. And that is the fact that for a long time, there was sort of a discrepancy, a large discrepancy between the flux that's measured at 100-meter depth and 1,000-meter depth that couldn't quite be explained, right? It's clear that the flux will decrease as water depth increases, but the decrease was much larger than could be explained. And there were various hypotheses put forward for why that might be, but nobody was able to answer it satisfactorily. And so what Nathan and his colleagues did was look at one hypothesis in particular, and that's this idea that large particles, as they sink, might fragment into smaller particles. And of course, smaller particles sink much more slowly. And so smaller particles will reside longer in the water column. They can be subject to microbial decomposition then. They'll basically be remineralized without ever making it to 1,000 meters. And that's important to know because it affects for how long atmospheric carbon is sequestered in the ocean, right? How deep that carbon gets transported. And so they were able to show that about 50% of this disappearance of organic carbon can be explained by fragmentation of large particles into small particles. So that means that this fragmentation is the single most important determinant of how far organic matter particles sink. Yeah, and it's a really important question for us to understand. So I hope these two examples sort of gave you an idea that tremendous opportunities come with these new observations. Let me talk a little bit about how far we have come toward this global target of 1,000 floats. So there's been good news a while back from our US colleagues. They have submitted a proposal entitled GoBGC, a proposal for funding to deploy 500 of these floats globally. And this proposal was successful. So over the next 5 years, there will be 500 of these floats deployed. And they actually still have funding to deploy 120 more in the Southern Ocean as part of this program that supported Alison Gray's study, which I showed you 2 slides ago. So we have here 620 floats that will be added to the ocean here over the next 5 years. And then there's also stuff going on in Canada that I want to tell you a little bit more about. So taking a step back, Canada has been a committed contributor to this Argo program since the inception, since the early 2000s, basically. Canada has had a sizable component of these physical Argo floats and continues to. And in 2017, a group of us got together, here you see a picture from that meeting, and talk about what we would like to do for BGC Argo. And an outcome of that meeting was a white paper, a vision paper that you can access here if you're interested. It's a very easy read, right? It's not highly scientific. It's meant for the lay person. We also have a website here about Canadian BGC Argo activities. And so I just have a sort of short summary here of some of the things that are going on in Canada with regard to these BGC Argo robots. Laval University has some really exciting work going on. They have sort of taken the floats to the boundaries of their capabilities and deployed them under ice and have had quite a bit of success with that. So again, right, this is really exciting to get continuous observations under ice of these biogeochemical variables. Then the University of Victoria has been adding oxygen sensors to a lot of the core Argo floats that DFO is deploying in the Pacific. So it's increasing our availability of, or the availability of oxygen observations. Then recently, the Ocean Supercluster Initiative has announced funding for a company, a Canadian company, RBR. They build sensors for this company to build 2 prototype BGC floats in collaboration with Dalhousie and DFO. And this is exciting because at present, there's really only one sensor manufacturer that supplies all the sensors that are used on the BGC Argo floats. And that's not very good for competition, as you can imagine. So the BGC floats are quite a bit more expensive than the core Argo floats, partly because one company has the monopoly. And so this Canadian sensor company, RBR, is sort of, interested in offering a second option here for scientists to purchase floats. So this is quite exciting. And then the last bullet here is about a project that was just funded that will allow us to purchase 42 BGC floats for deployment in the North Atlantic. And I'm going to tell you a little bit more about that. So I'm the lead of this project. It's a co-lead is Utapaso from Memorial University, and partner institutions are Laval University, University of Victoria, and McGill. And of course, DFO, our government partner. And there are 42 floats foreseen in total. 32 of these will carry the whole 6 sensor suite, and they will sample at the standard Argo mission that I described at the beginning, right, where the float drifts for about 10 days and then measures one profile. But then we also have 10 floats reserved. We'll have them probably with a lighter sensor load, partly for financial reasons. that we will use for process-oriented sampling. So by that, I mean, we will use them to profile more frequently and get sort of a different set of insights into processes. Also included is money for high performance computing infrastructure, because we want to develop models that will immediately ingest these observations as they become available and give us better estimates of what's happening in the ocean, right? Sort of turning these raw observations into products that are useful for any stakeholder, anybody who has an interest in understanding what's going on. And so there's a range of science questions we're interested in. The sensitivity of carbon sequestration and ocean ventilation to changes in stratification, for example. And I already showed at the beginning that this region here is very important for the uptake of anthropogenic CO2. It's also really important for supplying oxygen to the deep ocean. It's sort of a region where oxygen gets to the deep ocean that then is important for the whole global deep ocean. We're interested in understanding the changes in ecosystem productivity, oxygen loss, and acidification, right? The rate of change. And we want to assess climate models in the region. It's such an important region. I showed you that the climate models really don't seem to be getting the conditions very well. So by having more observations, we hope to improve them. And sort of a more infrastructure related set of objectives is we want to derive approaches for obtaining rate measurements. So not just concentrations, but also the rate at which properties change. So for example, the productivity, the rate of productivity, the rate of vertical export. We want to develop algorithms that extend ocean color observations into 3D, into the vertical. And we want to build this predictive system that I mentioned already. And so this graph here shows you sort of what we project will be the availability of profiles from this program compared to other currently available sources of observations. And you can see that just within a few years, this will be the most important. This light blue corresponds to this project here. This will be the most important source of oxygen, nitrate, and these other observations in the region. All right, and so I hope I've given you sort of a sense for the power and the power that these floats will have in helping us to better understand the changes that are going on. And sort of the idea is that it will allow us to address fundamental science questions, but also applied needs. And so it's very open to anybody interested in getting involved. The data is freely available, as I mentioned. So it's an invitation really to get involved. And I'm listing here some of these important questions that we can't properly address at present, given the lack of well-resolved observations, right? Ocean carbon uptake as the ocean warms, the deoxygenation, so the loss of oxygen, a quantification of variability and trends in ocean acidification, how primary production is changing as the ocean is warming, then the biological carbon pump, right? Really how effective it is, how long is carbon going to be exported. And then there are very applied sort of objectives. Marine management will benefit greatly from better assessments, and global carbon budgets will improve if we are able to more accurately quantify the uptake of carbon by the ocean. And so there are a lot of people involved in this who I should thank. There are, of course, current and former students in my group, especially Chris Gordon and Bin Wang, who've worked on these data, and colleagues in the Department of Oceanography at DAL, especially Marlon Lewis, who I mentioned earlier. Then internationally, Ken Johnson is our colleague from the US who's leading the efforts in the US, but also the global efforts, together with Hervé Claustre from France and other members of the BGC Argo steering team. Then in Canada, Blair Green and Denis Gilbert and Howard Freeland all three have had a really important role to play in making sure Canada is a committed contributor to Argo. Then, you know, all the members on the BGC Argo committees, all the co-PIs on this proposal that I just talked to you about, our funders, CFI, DFO, OFI, the Ocean Frontier Institute, and NEOPAR, and many partners. The logos are shown here. So I'll stop here and see if there are questions.
Okay, thank you, Katja. I'll do the honorary clap as I have done for your other talks. So I believe some people have been sending in, or at least one person has sent a question in by chat, but because it is only one person and because I actually know them, I'm going to ask Len to just ask the question in person. So Len Barry, if you could unmute yourself and ask away.
Hello, thanks. Can you hear me?
Yes.
Yeah, fascinating talk, wonderful. It's great to see the development on the ocean side of the Argo program. My question pertains to the overall international management and quality assurance of the Argo program, of the traditional variables and the future. Through what mechanism internationally are you coordinating this?
Yeah, that's a great question. So it's really quite well developed. And of course, PGC Argo is taking advantage of these 2 decades of Argo. So there are several, each nation that has a significant number of floats in the ocean, so take Canada, has a National Data Assembly Center, or DAC. So the Canadian one sits in Ottawa, and it's funded by DFO. Mm-hmm. So they perform both initial quality control and also what's called delayed mode. So there's an initial quality control that happens when the data comes in before it's pushed out to the world and publicly available, with the whole range of quality flags attached to it, all very well documented. And then there's sort of a delayed, a certain time period in which some sort of mistakes or errors that crop up from time to time can be corrected, really with involvement of a person looking and identifying any problems. So that's very well established. And so the data from the National Assembly Center are then pushed to 2 global data assembly centers who are mirrored. So there's one in the US at Scripps and one in France. Yeah, so the data is mirrored, you know, for redundancy reasons. And in terms of coordination, there's a committee where all the different data assembly centers, the national centers, have representation. In fact, the annual meeting is next week. There are different science steering teams, and there are the data management teams, you know, with membership from all the different nations. And the protocols are discussed. Any changes are discussed and scrutinized before they come into effect. And so with the biogeochemical Argo, we're a little bit behind, but it's the same model that's going to be applied pretty much. So does that sort of answer it? Or
yeah, is the UNESCO International Ocean Commission involved here? Is this sort of the large scale overall umbrella for this?
I don't think so. I think this really grew out of I think this is really more a bottom-up effort that grew out of the need of scientists to have, basically the impetus initially was to have accurate information even about the ocean heat content. And but it has gained international traction, right? It's supported by national governments around the globe, including Canada's. I don't think it's formally, but to be honest, I don't know exactly. If Blair Greenan is on the call, he's actually the Canadian Argo representative. he might know, he might know better. But it is certainly, yeah, it works very well. And
yeah, I'm just asking out of, in parallel with the atmospheric measurements, the meteorological services around the world coordinate their measurement programs through the World Meteorological Organization. And I understood that the IOC was of UNESCO was the equivalent for the ocean side. And I wondered if that was playing a role, or whether it's more on, sounds to me like it's coming more from bottom up research university groups that have fed into NOAA and DFO and organizations like that in government, right?
certainly true for the BGC side.
Yeah. Okay. Thank you.
Okay, thank you, Len. Thank you, Katja. So I believe Philippe Tortel has the next question.
Yes, thank you. Hi, Katja. Thanks for an excellent talk.
Hi, Philippe.
Hi. I wonder if you could just comment about the current state and challenges around sensor calibrations and drift. I'm thinking about optical sensors, for example, which have real problems with biofouling. I know they park at depth, so that's helpful. But what's the state of play there for the various sensors?
So biofouling is an interesting topic. Yes, it turns out to not be a problem for the Argo floats. They spend enough time at depth that it's not a big issue. Certainly not over 3 to 4 years based on experience. So, I mean, one thing that has been done is sort of profile floats with bio-optics on a 5-day cycle rather than a 10-day cycle. So you get more profiles over a shorter period of time. The intention was to mitigate potential issues of biofouling. So biofouling is not a topic that comes up in the discussions. It doesn't seem to be a huge problem. Now, other sensors, so oxygen is one that is known. There are two sort of specific problems with the oxygen sensor. One is that it might drift over time. It's basically the lamp that's used in the sensor, losing its power over time. Another issue is that the response time of the oxygen sensor is a little bit slow. So if you measure consecutively up and down, the oxycline will be offset between the up and the downcast. However, this can be corrected for. So this hysteresis effect can be corrected for. The sensor drift, actually the solution that has been developed to deal with that is that the oxygen sensor is now on a stick. It sticks out of the water, so on a stick sort of above the float body itself. So when the float is at the surface, the oxygen sensor sticks out and measures oxygen in air. Not because that's a particularly good way of measuring oxygen in air, but because we know the oxygen, the partial pressure of oxygen in the air quite well. So it can be used to detect any sort of long-term drift and correct for long-term drift if it is occurring. So that's the status with oxygen. So pH is the youngest sensor. I'm not quite sure what the challenges are with that sensor in terms of drift. I know response time is a bit of a concern there as well. But again, judging by our experience with oxygen, if we can characterize the time constant for this response time delay, we can actually correct pretty well for those errors. And nitrate is another sensor where we don't have that much experience with yet. So I'm not quite comfortable talking more about nitrate, but I'm very keen on learning more about it as we deploy these. Yeah, I mean, light is very well established as a measurement.
Okay, thanks for that overview. It's helpful.
Yeah.
Okay, well, I think we're very close to the end. So let me just read a couple of things that were put as comments into the chat. So Denis Lefebvre noted that the decrease of the oxygen content is one of the most scary parts of the outlook. And then to answer a previous comment, Dave Hébert noted that I guess the network is part of Ocean Ops, and formally and part of JCOMM. So if that means something to you, doesn't mean much to me, you know, feel free to look in the chat and have that kind of fill out the question period for you. So we've had a good hour and a great sort of set of talks for the year. So I guess maybe let's thank Katja one more time. And
thank you very much for listening. And those of you who ask questions, thank you for engaging.
And I hope, you know, we can continue to have these sort of very public events as part of what CMOS does. And actually we do have some stuff on our YouTube channel already, so you can have a look. If this interests you, there might be other stuff that you're interested in as well. And it's very exciting to see the science. And happy St. Patrick's Day, everybody. And hopefully next year we can, you know, go to the pub and have a pint or something like that. All right. Take care, everyone. Thanks again.